Okay, so this is my part, facilitated diffusion. Okay, so what's the definition of facilitated diffusion? Okay, it means a diffusion of molecules, ions, across the lipid layer along the concentration gradient through a carrier protein. You can see it right there. And it can be called as passive mediated transport because it doesn't require any chemical energy from ATP hydrolysis. Okay, so you guys will ask how to transport proteins facilitate diffusion. Okay, a concentration gradient occurs through, okay, so let's say outside there's higher concentration than the inside. And these molecules, they try to diffuse through the cell membrane, but it got repelled by the hydrophobic parts of the cell membrane. So that's why facilitated diffusion has the integral proteins that allows the molecules to actually bind or to actually pass through a carrier protein. Okay, so there are two types of proteins. The first one we call here is channel protein, and the channel protein contains a part, as you can see here, it's directly passed through the protein. And the cells actually pass through at high flux rate, which is a high speed. And it can transport only water or certain ions, and it's very specific compared to the one we call here is carrier protein. Okay, for carrier protein, the molecules actually have to bind to one side of the cell membrane, and after that it undergoes a conformational change. After the conformational change, as you can see here, it binds to one side here. After that, it changes the shape and allows it to pass through the other side of the membrane. So this is the carrier protein, and it acts like an enzyme. The mode of attachment is like enzyme substrate. It's something like that. Alright. So I'd like to give an example of facilitated diffusion, which is glucose facilitation. Okay, as we know, glucose is a sugar molecule that serves as the fundamental energy source for many cells. So outside the cells, which is in the bloodstream, there's high glucose. So inside the cell, the glucose molecule is low because of the continually consumed by cell metabolism. Alright, so this results in a higher concentration of outside the cells and a lower concentration of glucose inside the cell. Okay, so as we said, facilitated diffusion, it occurs along the concentration gradient. The glucose molecules actually tries to diffuse through this plasma membrane, but the problem here is the glucose molecule is too big to pass through the plasma membrane, so that's why it needs those carrier proteins in order to pass through the membrane. Yep, so that's my example. Next slide. Let's go back to it.
Hi everyone, I'm talking about filtration across the pattern gradient today. So I'm drawing a structure now. This is a structure of glomerulus, and as you can see, blood enters in these capillaries. This capillary is called an arteriole. Efferent arteriole. And the blood, which is leaving out the capillaries, is called an efferent arteriole. filtrate diffuses in or out of the capillaries is there are three pressure we need to concern about. The first pressure is hydrostatic pressure. The second pressure is the oncotic pressure. And the third one is the net pressure. There's two hydrostatic pressure. The first one is the hydrostatic pressure of the blood, and the second one is the first one is the hydrostatic of the blood. Hydrostatic pressure of the blood, and the second one is the hydrostatic of the filtrate. Hydrostatic pressure of the filtrate, and the third one is the oncotic pressure. So to find out the net pressure of these glomerulus, why do we need to find the pressure of this glomerulus structure? Because by determining the total pressure of this structure, we can know the filtrate will diffuse in or out of the blood. So how do we calculate the net pressure? We use the hydrostatic pressure minus the oncotic pressure. Let's say the hydrostatic pressure of the blood is 55 millimeter mercury, and the oncotic pressure is 30 millimeter mercury, and the hydrostatic pressure of the filtrate is 10 millimeter mercury. So you use the hydrostatic pressure, which is 55, minus the oncotic pressure 30, plus the hydrostatic pressure of the filtrate, which will diffuse into the blood, which is 10. So the sum of the net pressure will be 15 mm mercury. So because the value is positive value, therefore the filtrate will diffuse out from the capillary into the Bowman capsule. That's my part.
Now let's talk about another topic, which is somatic cell division. I'll be explaining about the interface part of the somatic cell division. Actually, the somatic cell division is a cell cycle, which is there's a series of biochemical events happening, leading to the cell's division and its DNA replication to produce two daughter cells. And then, for the cell cycle, there are three phases. The first one is the interphase. The second one is the nuclear division, which is the mitosis, and then the third one is the cytoplasmic division, which is the cytokinesis. And then for interphase, it can be further subdivided into three stages, which is the GAP1 phase, S phase, and then the GAP2 phase. Okay, now GAP1 phase abbreviated as G1. In the G1, the cell will regrow. The cell will regrow to its normal size following the previous cell division, and it will then also synthesize protein actively and the mRNA in order to prepare itself for the DNA replication in the S phase. And the cell will also carry out other normal cellular processes. Okay, now let's go to the S phase. Once the cell has received the signal to divide, it will then duplicate its chromosome via DNA replication, which has been explained by group 2 earlier. And so this process here is very accurate and precise in order to prevent genetic abnormalities in the new cells formed. So this process is highly regulated. And the last stage in interphase is a GAP2 phase. The most important checkpoint in this phase is the newly replicated DNA here will be double checked for errors. If let's say there's errors, the cell cycle will arrest, and then the errors will be repaired and to remove the errors. If let's say the errors are too severe, the cell will initiate apoptosis, which is the cell self-destruction mechanism, or often known as programmed cell death. And so if there's no errors to the DNA, to the duly replicated DNA, the cell will continue to grow and then will continue to synthesize more protein as a final preparation before proceeding to mitosis. Okay, thank you.
After the interphase, the prophase will occur. The chromosome contains the X-shaped structures, and each chromosome is composed of two sister chromatids, and which containing identical genetic information. Then the prometaphase will occur, and the nuclear membranes will break apart, and the spindle will start to interact with the sister chromatids, and with the chromosome. Sorry. Then metaphase will occur, and during the metaphase, the chromosome will line up end to end along the equator, and the central are now at the opposite poles of the cell, which with the spindle fibers attached to the sister chromatids. Then I'll pass the time to you.
Okay, after metaphase, here it comes to anaphase. During anaphase, we have spindle fiber. They contracts and shortens, pulling two sister chromatic to each side of the pole, separate themselves. So after that, here we come to telophase. During telophase, we have our sister chromatids. So nuclear embryos will form at both sides of the sister chromatids. Then chromosome will uncoil and uncondense. And next, two diploid nuclei will be formed at each side of the centroids. Okay, after that we have our cytokinesis. In order to form two identical cells with the parent cells, we have the process cytokinesis is to separate the two nuclei into two separated cells by forming a cleavage bureau between them. The microtubule in the plasma membranes will pinch to inwards, like this one. Pinch inwards, forming a cleavage bureau. Then two separated cells will be formed until the plasma membrane is separated. Yeah, that's all.